I was introduced to Nick Lane's work when I was in grad school, working on bacterial metabolism, and I had big questions about how life formed and what that meant for the experiments that we were doing. And my advisor gave me a copy of The Vital Question. And The Vital Question was basically a philosophical exploration of a different way of looking at the origin of life, because you have the RNA world option, you have the DNA world option, you have Panspermia, and then you have Nick Lane, who's like, look, the environment and life are fundamentally related to one another. And if you are going to look at the emergence of life on a planet, you cannot simply look at the biochemistry of life. You have to look at the biochemistry of life in the context of the environmental structures in which these cells could have emerged. And that was kind of mind-blowing to me. And Nick Lane has gone on to write more about the foundation of energetics for life, where, you know, the difference between life, a living organism and a dead organism, is not the genetic code. Both bodies still have the genetic code. It's the fact that the reactions cease, the flux of material from the outside world to the deepest core of the cell ceases. And what does that mean?
So we're bringing in electricity to the conversation in a very serious way today, and I think you guys will really enjoy that. If you have thoughts, please leave a comment and let's talk about it. And if you really enjoy the conversation, share it with somebody, bring them into the conversation, and hopefully we can move forward with all of this. If you really love what we're doing, consider becoming a patron at patreon.com/demystifysci, where you can join in our weekly discussions and really help us steer the ship as to what comes next. We want to take this show on the road. We want to be able to do live podcasts. We don't quite have the equipment for it yet. So even if you can only give a couple bucks, that's going to be a huge, huge help to us.
Our second camera currently only lasts halfway through the filming.
You may notice that.
You may notice that we stopped showing the other shots. So money for a camera. It would be good.
Otherwise, this was an extremely stimulating conversation. I think you're going to love it. So enjoy.
The scientific revolution starts now.
The Demystify podcast.
Let us see in the dark.
In some strange way, I'm studying what I've been working on for decades, which is to say how energy flow works in biology, how mitochondria work, the power packs in cells. And they work in a weird way. They've got an electrical charge on the membranes. And the machinery that's used to generate that charge is amazing and fascinating. The astonishing thing is it's basically universally conserved across all of life. I've heard it said, I don't know if it is strictly true or not, but that it's as widely conserved as the genetic code itself. So cells work this way. And so there's an interesting question, why? And the way I started answering that question, why, was actually through writing books, which, as you say, is bringing in not exactly the philosophical element, but asking why is permissible in books in a way that it isn't really in scientific papers. So...
Do you mean that science papers are asking how, like mechanistically, how something happens versus the underlying... How does why different from how? I'm sorry to interrupt you. I wanted to follow.
Well, I tend to confuse the two, so maybe that's the problem that I have. Why this way rather than how could it have been this way? Or to me, they're very similar questions. But for example, to a biochemist looking at a metabolic chart or looking at the enzymes in that chart, the question is, how do they work? What is the mechanism by which this enzyme catalyzes this reaction? And to me, the question is, why is it a rotary motor?
Why this one? Why this mechanism versus any of the other millions of or infinite possibilities?
And that's kind of a philosophical question, and it's. I think evolutionary biologists tend to ask why much more often than biochemists, and I've wandered into the deep evolutionary history of the planet. In the company of evolutionary biologists, but as a biochemist by training. And you tend to lose all your training as you go wandering around in the undergrowth. and pick up all kinds of other random bits and pieces. But the place you end up is asking, why is it like that? Why are all cells powered by an electrical charge on the membrane? And in a way, back to a how question, how did it come to be like that?
It seems like a particularly difficult question in a situation where n equals 1, and that we have one planet to look at with one life, one tree of life, I guess is the way that we think about it. Seems like the why question might come into more focus when you can see multiple planets and compare evolutions in each of those processes.
That may be so. We don't have that luxury yet. But n is 1 in a trivial sense, I suppose. If we break up the tree of life into bacteria and archaea, and the eukaryotes, which are our own kind of cells, there's a few strange things about that. The bacteria and the archaea look pretty much the same. If you look at them down an electron microscope, they're practically indistinguishable. But their membranes are very different in their chemistry. The mechanisms by which they replicate DNA is really different. There's practically nothing conserved between bacteria and archaea. So you have these profound differences in the biochemistry and the genetics between these groups, and also some profound similarities. They share the genetic code, they share the electrical charge on membranes and so on. So yes, n equals 1, but you've got this divergence which is really deep and about some of the most fundamental aspects of life. And so, and again, we have a 4 billion year history of life on Earth, and our own kind of complex cell, the eukaryotic cell, arose just once in that whole extended period of time. Now, it may be that complex life arose on hundreds or thousands of occasions. You can't say it didn't, but there's no evidence for that. So, everything that we can see, everything we've ever found, falls into these 3 domains of life. And viruses are kind of slightly separate from that. So it may be that all of this is turned on its head tomorrow as we discover something else, or it may be that it's really telling us something. And what it's telling us then is that it's genuinely difficult to evolve complex life. Now, the reason I think that's the case is that there was an endosymbiosis involved. There was effectively some cells got inside other cells. This was originally proposed decades ago by Lynn Margulis, and actually went back even before her. And effectively, it changed the topology, the structure of cells, the way that they operate, the kind of the mechanics of cell physiology, and allowed them to explore a different genetic sequence space, and you kind of end up with a different place in evolution. So then the question is, well, what was the constraint? If it's about the electrical charges on the membranes, do cells need to have electrical charges on membranes? Could it have been quite different on a different planet? And then you're left back with these effectively astrobiology questions about what does life have to be carbon based? Does it need water? Should it be cellular? Will it have electrical charges on membranes, and so on and so on? And yes, although n is 1, we can explore these questions. There's a very good reason why life is carbon-based. It doesn't mean it has to be, but carbon is very common, is very good at what it does. And if we find 1,000 examples of life, it wouldn't surprise me at all if 997 of them turned out to be carbon-based, because it's so common and it does what it does really well. But as soon as
it seems more than carbon, even they're water-based. Like, water is such an important part of the picture, and if some other solvent was capable of doing the job that water was, I'm not convinced that that's the case. But I mean, it seems like there's other possibilities for how something could happen, even with carbon itself.
Yes, and people talk about Titan, for example, where you have liquid methane, and carbon chemistry in liquid methane is almost inverted, because anything bigger than a handful of a chain of more than about 5 or 6 carbon atoms doesn't dissolve in methane, and so you end up with, if you wanted to have a cell structure, the membranes would be soluble and the insides would not be soluble. It really confounds the way that you would think about what life might be like. But the more you think about those problems, the more you realize, well, water's actually, again, really good at what it does. And we don't really know what it does, because we don't really understand how water works at all. And again, it's really abundant. So So the likelihood is we're facing carbon, we're facing water. Cell structures are not only important because they keep the insides different to the outside, but they're also where all the work is done across the membranes. And again, that's about the difference between the inside and the outside and the power. So you find yourself generalizing, maybe spuriously, but generalizing back towards why life is this way here. And you begin to think, well, if we find life elsewhere, it wouldn't surprise me very much if it looked a lot like bacteria.
Yeah, that's a really interesting perspective, because people are always imagining that aliens would look completely different than us. And yeah, if you take the idea that life can only happen one possible way, that everything, that the answer of the why is because that's how it has to happen. That's the way that
it has to happen that way. It's more probabilistic than that. It's that carbon is abundant. It does this really good chemistry. It does it especially well in water. You've got this beautiful mixture of soluble and insoluble components. So you can have membranes, you can have compartments and so on. And all this happens beautifully in water. It's probably possible for it to happen in other solvents as well. But again, water is so common that the likelihood is going to happen in water. It's also noticeable that on Earth, carbon is about the 12th most abundant element, and yet, silicon is considerably more abundant, but there isn't any silicon-based life here that I'm aware of, anyway.
Well, it's interesting because what I immediately think of is on a hydrocarbon-abundant planet, you would have cells that are maybe single bilayer rather than or a single layer versus a bilayer membrane, where what you have is you have basically this bubble of things that are soluble, and they communicate. The reactions of life are happening inside of that vessel. And whatever transport is happening by like sending out little blobs of water into this hydrophobic environment. And that doesn't seem impossible, because water would organize quite neatly into, contained vesicles. And
it's possible to imagine it. Whether it would work all the way down, I don't know. Could you build a molecule like DNA that way, an equivalent to it? I don't know. I've not really thought about it.
But you're working on this kind of stuff in your lab, right?
Kind of the level of hydrophobicity, or whether it's mixing with water or soluble or insoluble. Yes, you can picture it. But whether you would be capable of building large, complex machinery that works, I don't know. It's not something that
it really seems like water is wholly unique as a molecule. Like it can act as an acid or a base. It can hold tension under different situations. The ice, there's like, I don't know if there's hundreds, but dozens of different ices that can form. It's just hard to imagine. It almost seems to me that humans or life forms are water, which has organized a bunch of molecules into behaving themselves, because 99 out of 100 molecules in your body is water, and they bear tension. They work to push against one another. They separate molecules. They selectively solubilize molecules. I mean, it's like what else could possibly? I guess I'm trying to I kind of agree that it seems like this is the way things probably should occur.
My
probably
goodness.
Might occur. I guess I would say it seems like water is how it has to be, but I understand that the more probably the more cautious way of putting it is probably.
Yes, I suspect water is fundamental. I imagine it's possible that it's possible to do it other ways, but far less likely because it's far less abundant, and it's we know all this carbon chemistry works. The thing that surprised me most from my own work and work going on in the field on the origin of life over the last 5, 6, 7 years or so is that if you start with the kind of molecules that were freely available on the early Earth, hydrogen and CO2 and so on, and you drive the chemistry, I'm thinking under hydrothermal conditions, but it doesn't have to be that way. What you end up with is the basic core biochemistry of cells. You get initially what are called Krebs cycle intermediates, but these are basically the kind of the standard hand of cards for carbon-based chemistry. There's just carbon, oxygen, and hydrogen. And everything else is made from them. In bacteria, in archaea, the chemistry is more deeply conserved than the genes themselves. So you see a divergence in the genes in the bacteria and the archaea, encoding the enzymes that catalyze the same pathway. So different enzymes catalyzing the same basic underlying pathway. And that pathway, or those pathways, seem to happen spontaneously in the lab in the absence of any enzymes or genes or anything. So again, there's an organization which happens in water, which happens with CO2, which happens at certain temperatures and so on. You can imagine it happening other ways, but it's almost uncanny the extent to which, from a certain starting point, the chemistry of life just unfolds.
So much of this chemistry is harnessed to an electrical system, right? So metabolism, you have the Krebs cycle, which I think we should talk more about it because it's so central to Transformer. But to kind of give an overview, you have this cycle that is passing along intermediates to proteins that are embedded in the membrane of the mitochondria, which then use those substances to catalyze the translocation of protons across a membrane. You have a reservoir of these protons, and then they fall down through this ratcheting rotary mechanism that produces ATP, and then ATP is sent out into the cell where it's historically been viewed as sort of like the gasoline of reactions, but I think that I think you are actually the one who started to point out the fact that that's not quite right, and that it is behaving less like something that's being burned and more like something that's aiding a reaction.
Yeah, I mean, I think we've all been misled a bit by ATP because it's so central to us and to animals and to cells generally. But this is something that's bugged me for a long time. I mean, the ATP synthase is this motor that sits in the membrane, and it's literally a rotating motor. It's a beautiful device. It's a nano machine, and so it's universally conserved across life. There are a few things have lost it, but pretty much everything has had it at one time or another. And so the question is, well, where did it come from? How did it arise? Can we think of prebiotic intermediates to get us along the pathway to that? And I've never been able to think of anything. I can think of genetically encoded intermediates, but that's still assuming genes. What before we have genes? And it's refocused my mind on what are the essential moving parts here. And the essential moving parts include the Krebs cycle that you mentioned, and it includes the electrical charge on the membrane. But it doesn't include the ATP synthase. And it doesn't include ATP either. So ATP, I mean, it's usually called the universal energy currency, and I tend to think of it as like a coin, really. And a protein, like a slot machine, you put the coin in the slot machine and it does something. There's some mechanical change in the conformation of the protein. And then it reverts to its position. So you put another coin in and you keep doing that. It just does one thing and it keeps changing its position. And that's what's driving everything. But the question is, we're turning over the ATP. We make our own body weight in ATP every day, pretty much.
Whoa.
So we lop off the final phosphate group. That releases a little package of energy, which does a conformational change in the protein. And then we use the power of respiration to put that phosphate back on again. So we're not synthesizing the entire molecule, we're just chopping the end off and sticking it back on, chop the end off, stick it back on. But the power of respiration, what we're doing with the Krebs cycle, in effect, is we're breaking food down to these simple molecules, the intermediates of the Krebs cycle, which are made of just carbon, hydrogen, and oxygen. And then we're stripping out the hydrogen, and we're stripping out the CO2. And that's basically it. We're not stripping out the hydrogen as hydrogen. It's actually as NADH, but it doesn't matter. Just think of two hydrogen atoms coming out. And the CO2, we breathe it out. And the hydrogen, we burn it in oxygen in respiration. So that's where all our power is coming from. But we're not burning it in a conflagration or something. What we're doing is splitting the hydrogen atoms into 2 electrons and 2 protons. And the electrons effectively are passed along almost in a wire of proteins within the membrane itself. So we have a current of electrons to oxygen. And that current of electrons powers the extrusion of protons across the membrane. So you mentioned a reservoir of protons on one side. But that reservoir, because they're positively charged, is also electrifying the membrane. So now we've got both a concentration difference, because there's protons on the reservoir of protons over here and not on this side, but we also have a charge difference, a potential difference across the membrane. And that's the thing which is universally conserved across life. You get rid of oxygen easily. You can put something like nitrate there, or you can put sulfate there, or you can even put carbon dioxide there. And you can still generate the charge on the membrane. So the common denominator is this electrically charged membrane. And it's not the protein machinery that's generating the charge. And then the Krebs cycle is a common denominator as well, but not as the cycle that we know it. What we see in some of the most ancient bacteria is that they do it in reverse. And so, as I said, the Krebs cycle pulls out hydrogen, pulls out CO2, and effectively generates a charge on a membrane. And what happens in some of the ancient bacteria is they're using the charge on the membrane generated in other ways to power the reaction between hydrogen and CO2 to make the molecules of the Krebs cycle, which are then the basis for making the amino acids and the sugars and the nucleotides and everything else. So it's gone from being a kind of an energy generating mechanism. It started out almost the opposite. It started out as a kind of a bodybuilding mechanism. It started out as the machinery for biosynthesis, really, the engine of biosynthesis.
So this leads us really neatly into one of our favorite topics, which is the question of electricity. And this is something that I ran into as a hard stop when I was doing my thesis, which is that biologists get to the point of recognizing that there is this electrical series of events that are happening. You know, we even talk about the metabolic machinery inside the membrane as a wire. What is electricity? Like, what is charge? What does it, right? Like,
I think it's actually a really important discussion for the biologists to get involved in, because it seems like once we understand what is physically occurring, like materially with respect to the atoms during these charge transfer events, obviously they're important in constructing new compounds and dissociating the compounds and providing juice to the system. But I feel like once we understand those basic material questions about the concept of charge, maybe it will make more sense how it comes to be or how life even comes to be, why this is a favorable situation, why it's something that should self-organize. Because otherwise we're like stuck looking for an organizer. It's such a strange paradox being that the abiogenesis question is just so against all of the dogma of basic science.
It's very godly.
It's very strange, yeah. It's a very strange assertion that life should come from no life.
I don't see it that way, I have to say. But it's a very confused field. It's confused not because the people in it are confused, but because everybody has their own idea, their own agenda, their own hypothesis, to put it in a more positive frame. And there's not much in common between these different hypotheses. Now, it may turn out that there's more in common than we think, but it's actually really difficult to say, oh, well, I'll have a bit of this and a bit of that. It makes you realize that to formulate a hypothesis and to test that hypothesis requires being quite purist about the hypothesis. It doesn't mean to say you have to believe you're right, but it does mean to say that you need to formulate this leads to this, leads to that, and so on. And the problem with the origin of life is that there's so many steps. I mean, I was thinking about how do I get this across? And I gave a talk in Geneva last week, and I ended up showing a slide of one of David Goodsall's pictures of E. coli with all the astonishing complexity of proteins and structures and DNA. They're just really, really stunningly beautiful as works of art, but also stunningly amazing as a depiction of what life kind of looks like, if you, in the imagination at least. And I found myself saying that the progression from simple prebiotic chemistry to that level of complexity is probably far greater. It's a much greater distance. It's harder to explain than going from a bacterial cell that you can't even see with the naked eye to a Tyrannosaurus Rex or something. It's the evolutionary distance to a bacterium is at least as great, if not considerably greater, than the evolutionary distance from a bacterium to dinosaurs or humans. And that's what's often forgotten, because people tend to focus on one aspect of the question. If you're a chemist, you think about the synthetic chemistry. Okay, the challenge is make nucleotides. So chemists go away and make nucleotides. They do the job. They started with cyanide, and the biologists say, hang on a minute, life doesn't really use cyanide very much. Are you sure you got the right starting point? And they say, yeah, well, we started with CO2 and it doesn't work really. So, go back to your own corner and you get on with the selection bit. But selection depends on what is being selected. And what is being selected depends on, well, do you think cells came early? Do you think protocells were early? Do you think naked genes came early with an RNA world? Do you think that membranes came early? Did metabolism come first? What order did these things happen in? And there's no agreement on that at all in the field. And I have my own ideas, and a lot of people would disagree with them. But the joy of this work on the origin of life is that it seems to be joining up. And the reason I say, from my own point of view, it seems to be joining up is that this metabolism, starting with CO2 and hydrogen, which 5 or 6 years ago, the chemists simply could have said it doesn't work and would have been justified in saying so, now cannot do that, or should not do that. Because it does work. It may not work with high yields, and it may not work in the way they would like it to work, but it works. You get all these intermediates. They're favored. They're along this pathway. So you say, how does it self-organize? And the answer is, in effect, what we start out with is a planet where all the electrons have gone to the center of the Earth, if you like, with the iron. They're all sunk down with gravity, with iron, into the mantle and into the core.
And what that in terms of chemistry, what that means is that the electron donors are down there, right? It's not like the electrons are separated from the iron. It's the fact that
I mean, there's no actual charge or anything. The iron is iron, but iron would like to be rid of its electrons and become oxidized and form rust, in effect. Mm-hmm. Whereas on the outside, we lose hydrogen to space. We have volcanoes, which are degassing at high temperatures, and mostly what comes out at those temperatures is CO2 rather than methane. So we have basically an oxidized atmosphere, an atmosphere which is hungry for electrons and wants to react. So oxygen will grab electrons from almost anywhere and become water. So the Earth then is a kind of a giant battery, where the inside is the negative electrode and the outside is the positive electrode. And the hydrothermal vents are the kind of the connectors between these two things. And the pores in the hydrothermal vents, so where we have the immediate mixing between one fluid, which is highly reduced, but basically hydrogen and alkaline, and the outside, which is oxidizing and acidic and with more CO2, not strongly oxidizing, but somewhat. And that's the same topology that a cell has, and it's the same topology that the Earth has, and it's the same topology that these pores in the vents have. And I find that rather graceful and elegant. It doesn't mean it's
and a battery for that matter.
Cells are batteries as well, for exactly the same reason. Is the positively charged outside relative to the inside. The energy for all of this is coming effectively from the flow of electrons then, and so from hydrogen to CO2. And life is, another gobsmacking thing to me really is that if you have a mixture of hydrogen and CO2 in a test tube with a lid on, and you heat it to 50 degrees centigrade, and you shake it up, that's less stable than if it all reacted to form cells. Thermodynamically, energy is released if those two gases react to form, total cell biomass. So everything, including the nucleotides. So thermodynamically, the reaction is favored. Kinetically, it's not. Kinetically, they're not going to react because CO2 is a rigid molecule. It doesn't want to bend very much. It doesn't want to react very much. And again, hydrogen is pretty stable. So there's a stability to these gases that means they won't react easily. And what biology is really doing, and what catalysts can do, and what the electrified membranes can do, is effectively break down the barrier to their reaction so that they will react and will form these organic molecules. And when they do that, it releases energy, which increases the entropy of the universe. It releases heat, and the heat increases the juggling around of everything else, and entropy rises. So, the origin of life increases the entropy of the universe.
Do you spend much time thinking about the flow of charge, the flow of electrons, like what that physically means? Because there's so many conflicting models of how that process unfolds, and these molecules, these I don't know, we call them organelles, but these respiratory structures, they act like wires to some extent, but they're very complex wires with switches and gates, and they have the ability to change the shapes catalytically of other molecules. Do you think much about what that means for an electron to flow?
Yes, and first thing to say is I'm not a physicist, but a lot of
the physicists don't know either.
are questions in physics. Yes, I think you're right. So what seems to be happening with the respiratory chain, and this is another interesting thing. So I say a wire. We have these giant protein complexes, but inside the protein complexes are effectively a bunch of standing stones, you might say, like crossing a river with from stone to stone. Now those stones are redox centers where an electron can sit. So the earliest ones are made of iron and sulfide. They're called iron sulfur clusters, and there's usually 4 iron ions and 4 sulfur ions in them, forming a little cage, and that's capable of picking up 1 or 2 electrons. And the next one is very often typically between about 8 and 13 to 15 angstroms away, that kind of range, which is the range for quantum tunneling of electrons. So no one, to my knowledge, has ever explained why. So in complex 1 of the respiratory chain, there are, I think, I can never remember the exact number if it's 7 or 9, but there's a bunch of these anyway. Why? We have a molecule docking over here, and we need work to be happening over there. And in between, we've got a bunch of stepping stones, and electrons effectively hop by quantum tunneling from one to the next. The way I read that as a kind of bad physicist is, well, it depends on the distance more than the actual reduction potential of the center. So if there's an electron here, then there's a fair chance it's going to hop to the next one, so long as it's within about 10 angstroms. And if it's on here, then there's a fair chance it will hop to the next one, so long as it's a similar distance away, which means there's a certain probability that electron could be anywhere along that chain. And that means if you put an electron on at one end, you don't know where it is really in terms of quantum indeterminacy. It could be anywhere along that chain. And presumably the longer the chain is, the more lost it can be. Which means that you can modulate the likelihood of an electron being anywhere in the chain by modulating the length of the chain. This is my imagination. I'm not really talking science here so much as
I think it's really important to imagine it. Yeah, I mean, my background, my PhD is squarely in material science and material physics,
right.
And we've been thinking a lot about this problem for many years now, and one way we found of looking at it that I think is really useful is, instead of treating electrons like these magical little currencies that plop from atom to atom, we think of it as a particular surface motion of the shell, the electron shell of the atom. And when in something like the electron transport chain, you have a number of atoms converging and interacting, you actually have a situation that's much like a gearbox in a car, where the surfaces are interacting with one another such that momentum can be transferred in a rotatory fashion from one end to the other very quickly, actually. And so it's not I think that we can start to think about it in a more mechanistic sense this way. I'm not saying it is so, but it's an interesting way of imagining the process unfolding in a way that doesn't involve the magical handing off of these blobs of God knows what to one another that can do special stuff.
We do know in terms of, I use the word current, and a current is another of these terms that I don't really know what a current in a wire looks like either. But in the case of respiration, the electrons are genuinely starting in the Krebs cycle on effectively hydrogen atoms in the Krebs cycle, and they are genuinely ending up on oxygen. So whatever these blobs are, they move over there.
The question is whether an electron is a material or whether it's a motion. If it's a particular discrete motion that is generated, then things start to make a lot more sense. But if we treat it like a material that's moving around, you run into a lot more problems.
And this is something that I was really grasping at when I was finishing my thesis, because there was no way of getting around writing about the charge in the membrane. Like, what I was studying is I was studying a redox-sensitive molecule in a bacteria that appeared to tie into the electron transport chain, right? Like, redox inside of a cell is this massive question. You look at a diagram of cellular metabolism, and cold horror grips your heart at the hope of understanding it.
I just wrote a book on it in the hope to kind of relieve the cold horror, but I'm not sure I succeeded.
I mean, I think that the book is good in the sense that it anchors you deeply in the way that the Krebs cycle at the center of it is what is feeding the various pathways of the cell and driving metabolism. But I think that there's still a sense when you look at it from an engineering perspective, where if you start to you mess with NADH levels, but then there's all of these other compounds that will react to that change, and the way that they react I don't think is predictable. We don't necessarily have very good...
I mean, I can't predict it. I'm working in the lab on that question as well, and every day my immediate prediction, it turns out to be wrong. It's an incredibly complex system.
Right, and so and that to me is a sign that we don't really understand it that well, because the minute that you can be like, okay, I will put this amount of this molecule or this amount of charge into the system, and this is the output that I'll get out, that's at the point where I throw my hands up and I'm like, okay, mystery solved, the cell is a machine, we understand how to run it. And like, something that just really, to me, seemed obvious about charge is that when we talk about it as this thing that it kind of floats around in the space of the cell, and it's almost this irrational thing, it is the equivalent of our perspective of the cell prior to David Goodsell's drawings.
And you go and ask the physicist what charge is, and the physicist will say it's it's a pair of opposites that exert opposite pressure, basically exert opposite pressure. And you're like, what is that? It's so abstracted.
Yeah, they do stuff and it flows and it moves and it's powerful, but you have the rotary motion of the ATP synthase. And so it makes...
And you got to remember that this is how we used to think of heat until very recently, maybe 150 years ago or something. We used to think of heat as this little magical particle thingy that moved around and did stuff. But we slowly came to understand that was a motion of the atom, right? And it turns out the accounting works perfectly if you treat heat as a little particle thingy. It works, you can pull off the same dynamical expressions using that model, and your technology will work and everything. You can have, but it's not so.
I think you're both. Sorry, go on. You were going to say something.
What I want to say is that there's something about the way that the chemistry works, where you're like, look, you take these electrons off of this C3 carbon that's inside the Krebs cycle, and you pass it off to oxygen, and at the end of the day, it comes down to physical contact, right? Because you can't just have that reaction happen in space and then transfer it to the oxygen. There needs to be this ability to move things into position in order for the molecules to resonantly connect to one another, right? You look at an iron-sulfur cluster, and this is a resonant antenna. There's no other way to see it. It's set up in such a way where you can imagine the iron at the center vibrating, basically like a trampoline almost, or like a drum. And then it passes it on to the next molecule, and that is what gets passed along until it gets to the end of the chain, where the oxygen is the final most receptive molecule for it. And I don't know that it's the same hydrogens that are in the inner membrane space that the oxygen is reacting with. It's just the fact that it floats in this bath, and the resonance of the oxygen is then what allows it to click into water.
We have no idea where those protons are. You said the intermembrane space. I would say probably the Christie space, because we now think of it as a closed space. But you can calculate, you can measure the pH, but pH is a bulk property in solution, and we're dealing with a confined space and extreme crowding and charged surfaces. And the answer that you get from using a pH sensitive dye or something is it says, okay, it's pH 8 perhaps in the matrix, and it's pH 6.5 to 7 in the Christie space. And so, well, how many protons are there then? And if you calculate the volume and the number of protons that would give you in that volume, the answer is, well, there's about 3 protons in the matrix of the mitochondria. And yet we are making our own body weight in ATP every day, and that requires a flux through the ATP synthase where we have 8 or 10 protons coming through to form one complete turn of the motor. So there's all these protons. We know that there's a circuit of protons going on, but if you try and count them, they're not there. So where are they?
And I think that's where, when you start thinking about the charge as a particular rotatory pressure between the surfaces of atoms that are in that material, then the opposite rotatory pressure is the opposite charge, obviously. And you can solve a lot of these problems where it doesn't appear to be material actually moving at the rate that's necessary to pull off the energy transfer.
Like with currents, like if you look at current, for a long time we had a model of current, which is the flow of electrons. Mm-hmm. But the electrons do not flow inside of a wire. And so our entire idea that what is happening is the flow of these little bowling ball particles inside the cells doesn't make sense. And then because that's the thing, you ask somebody in biology, what is a proton? Or you ask somebody in chemistry, what is a proton? And there's just this, like we've run into this before, right, Shiloh?
Well, I mean, constantly, yeah, of course. But this, I think, is just because we need to punch through this next paradigm. It's like the heat to kinetic theory transition. We're on the verge of something like this. And I think that it's quite apparent that the surface of the atom is undergoing this process. We call it spin. It's a particular kind of involuting rotation. And I think the atoms seem to have the ability to join shells with one another at close range. And you can imagine the performance of a great deal of work at a distance this way, just by linking everybody up. And anyways, the details of it are yet to be completely enumerated, but it's one of our pet topics that we love so much.
Right.
And
it's not something I know, but I mean, when you say what's a proton, I tend to think of the nucleus of a hydrogen atom, and I know it's not going to exist by itself. It's going to be, people say it's bound to water as a hydronium ion. Other people say, no, that doesn't exist either. It's a larger structure. Pretty much all the proteins that are on the Christie side of the mitochondrial membrane have a negative charge. And that negative charge, well, it depends, on technical things like, well, what's the propensity to pick up a proton? What's the pKa? But there's a huge buffering potential. So all those protons are effectively bound to something else and do not exist as free protons. But then you're talking about flows. There's some tendency, and I don't know how it works, that across this entire negative charge surface, there's probably a tendency of a proton to not be anywhere, to be somewhere smeared as across that whole space, which is how they can all end up in the end going through something like the ATP synthase.
And it works if by proton you mean positively charged region in this whole molecular soup, because you're talking about a motion then. You're talking about some momentum in the system that's being applied to the whole network such that you're actually driving it throughout the entire network.
And I think that biology is uniquely placed to be able to figure this out, right? Because it is... You talk in Transformer about the immense difficulty of determining the stoichiometry of these metabolic reactions, right? We're talking about people that have they're mashing up tissues and they're putting them in these devices where they can measure the change in gas pressure, and they're working in the dark, right? Like their hands are inside of a black bag.
That was, you're talking about Krebs and people, which was 80 something years ago. But we're doing the same kind of things now. We're doing it in a bit more sophisticated way, but it's basically the same stuff.
Right, because we can't get down to the core of what is actually there because we can't see it. And physics is, you have your particle accelerators and people are working in their own black bag over there, but I feel like biology is a place where the mysteries of physics are kind of laid bare because the cells have figured out how to do things technologically that we cannot. And so biochemistry lies at this interface where I think that it can give us deep insight into what is actually happening down there based off of the things that you detect where you measure for the protons and you're like, well, there's not the free protons there. And so what does that
and you can also measure you can use microelectrodes to try and measure what is the membrane potential. And people have done this since the 70s. And the answer is, with a direct measurement like that, it's almost not there. It's 10, 20 millivolts. You use the dyes, you use pH measurement, you can use all kinds of other ways of measuring it, and that's telling you, and also just the chemistry itself gives you a kind of upper value of 230 millivolts. And I will throw around this 180, 200 millivolts. Nobody's ever properly measured that. This is really a constructed number based on a lot of data and a lot of evidence, and it's not a make-believe number, but neither is it a straight measurement with a microelectrode. Why? Because the structure itself of mitochondria is far more sophisticated than we had realized. Why don't we know that? Because until very recently, you do electron microscopy or something, and again, you're doing it in a vacuum. You're chopping up tissues. You're producing, in effect, something which, the fact that it's not a complete artifact is amazing. The fact that you can see anything at all is amazing. We're getting better at it now with cryo-EM and so on. But again, it's a very invasive procedure. And to see the real, the living structures of mitochondria as they are.
And the structure of water too, right? The structures of water. I mean, I've got a paper in review right now where we're looking at, in a biological context, how water could play a role in tension signaling, which is just crazy. Like, if you think about David Goodsell, I love his work, first of all, incredible, but no waters anywhere,
Yeah. Actually, he did for my last book, Vital Question. He did a specially commissioned ATP synthase for me, and he did add some water in, not as structured water, just for scale, but he occasionally does. But yes, do we know? This is something I read a very thought-provoking book, The Fourth Phase of Water, by Gerald.
Pollock.
Yeah, we've had him on the show. We've had him on the show twice, actually.
Yeah, he's a good friend.
Okay. Yeah.
Love it. Yeah.
Yeah, no, it's a very, very interesting material. I part company with him periodically because some of the things he says I just don't agree with. But I haven't read a more thought-provoking book in a long time, because if he's even partly right, and I think he is, then the structures of water around membranes, around proteins, and so on is absolutely critical to how everything works. I don't think it means that that everything's being done by the water and that the proteins and the membranes are not important. But if you've got structured water along the surface of a membrane, then how are you going to pump protons across that membrane? I'm fairly persuaded that we're pumping protons across the membrane, but then you need channels. And you can bet your boots that biology knows exactly how to organize a water channel with hydrophobic surfaces instead of hydrophilic surfaces. And none of this is factored into how we think the system works. I mean, it's just not known. And most biologists are a lot more conservative. And as soon as you start talking about the structure of water or electromagnetic fields or, just conventional things in physics, most biologists will start to back away and think, okay, he's gone over the edge.
It's terrible, too, because the physicists back away, too, because they're like, hey, we have perfect mathematical descriptions, we can build technology. And you're like, yeah, they did for heat, too. And they're just like, get lost. Like, we don't need you here. And I don't know. I love Jerry Pollock, but he also has a way of talking in which it seems like these are not mainstream ideas, but the truth is they're extremely mainstream ideas, just not in biology. Like out in material science, like with the study of filtration systems and carbon nanotubes, these water structures are highly well developed. And, you can use atomic force microscopes and actually probe down onto surfaces and see different structured levels. These aren't like super far out ideas, although they haven't necessarily penetrated the biological disciplines. So
yeah, because that's the place where the in vitro versus in vivo really starts to become apparent, because you can take a sheet of mica and you can look at the structure of water with an AFM and you can see that there's this beautiful hexagonal layering of them on top of each other, and you can start to create these hypotheses about what that might be doing.
And it's patterned by the surface that it's on and so on.
But you can't get into a mitochondria to look at the inner surface.
You practically destroy everything getting there. So
yeah, exactly.
It's really difficult.
That's the worst thing about working with living things is they tend to die.
Yeah, they do tend to die. That's what I hated the most. But I think that biology is... It suffers a crisis where when students go into the world of biology, what they do is they learn rote memorization of this is how these proteins work, this is how the DNA works, this is what biochemistry looks like. You're going to memorize these reactions. You're just going to know a set of facts, and then what you're going to go out into the world and do is you're going to find the next little piece that can be folded into this set of facts. And maybe that's the case with many sciences. I don't know. I did a biology PhD, right?
Yeah.
And it was a very molecular PhD. And I think that what your work really strikes at is how much is still mysterious and unknown in biology, and how our ideas of the way that life works are still not quite well formed. Like the gene first, the RNA first model that Richard Dawkins really popularized, and then I think that it was it Woese who had the RNA world?
Woese, yes.
Yeah, Carl Woese. So these two ideas are central to the way that we look at biology right now.
Yes, and the gene-centric view has taken us a long, long way. I mean, it's exciting stuff, or it was exciting stuff. the discovery of the genetic code, but that was back in the 60s. And then beginning to sequence things and realize that all life is related, and we can track back a tree of life back to some of the most ancient bacteria. What we've learned from it is tremendous, and has revolutionized the way we think about things. But it's also very easy to do, easy in the sense of it's conceptually easy. All you need to do is get your stuff and stick it in a machine and it'll give you a sequence. So it's turned into kind of information processing, and there's reams and reams of data that you can play with for as long as you want, really. And instead of thinking, if you don't see a pattern, you just get more data. And all the money goes that way. Billions of dollars sunk into, first the human genome, which was a sensible idea, but then the epigenome ideas where is 80% of the genome functional? No, it's not. Probably 10% is functional, maybe 20 at the most. The rest, we don't know what it does, but it's definitely not. It's not functional in a conventional sense. Maybe it's functional in an unconventional sense that we don't know very much about. But it's certainly not under any long-term selection, because the mutation rate is unconstrained. So whatever its function is got nothing to do with its sequence. And so sequencing it is the last thing you should do then, because the sequence obviously doesn't matter. So I think the problem I have is not that this is all wrong or even misleading. It's all true. It's just that it's one bit of biology, and there's an awful lot of other stuff going on, and too many people have backed away from that other stuff, or decided it's irrelevant, or have just focused on the information aspects of biology, and without even questioning themselves, just assume that information comes first and that everything else follows on from that, and that genes effectively invented metabolism and so on and so on. And it's, they don't realize that's a huge assumption with a lot of implications that are directly testable if you're working on the origin of life, most of which fail.
And they fail because it's impossible to create conditions where the nucleotides or whatever chemistry you end up developing actually progresses to some kind of metabolic machinery?
Yes, I mean so this idea of an RNA world where you've got kind of naked replicators. First of all, you need to make them, which means you need an environment to give you bucket loads of nucleotides. So people working on an RNA world just assume that there's bucket loads of nucleotides out there. They're all just floating around in the ocean, and you need some, and oh, look, here they are, which is already a very questionable assumption. And then we polymerize them to make RNA or DNA or whatever it may be. And again, it can be done. So it can be done to make these, starting with cyanide. It can be done to polymerize them, having wet-dry cycles. You effectively, to join two nucleotides together, you have to pull out one water molecule. So it's a dehydration reaction. And it's obviously easier to do that if there's no water around. But again, that's not what cells do. So there's two difficult steps there. Now you end up with a population of these RNAs, and the whole basis of the idea is, well, they can both replicate themselves and they can catalyze things because they can fold into interesting shapes like proteins and become ribozymes, and therefore they should be able to catalyze their own replication. So all we need then is to find this magic RNA replicator that can make copies of itself and catalyze things. And people have been looking for decades and found quite sophisticated systems where this can make a copy of that, which can make a copy of this, and you go in a kind of a circle, and eventually everything can make a copy of the starting point. But the idea that this somehow now gives rise to all of metabolism and encodes all these enzymes and constructs a flux through it, there is not a jot of evidence that happens. No one's ever got anywhere near it. No one's really got anywhere near even coding for a single metabolic process with a ribozyme, to my knowledge anyway. So it's a million miles from where we need to be. And the alternative assumption that metabolism is coming first also makes some really big asks, if you like. It basically says that metabolism just happens, just like that. It's thermodynamically favored, and a long pathway with 12 steps in it that are normally catalyzed by 12 enzymes encoded by 12 genes. These intermediates will spontaneously interchange between them at very low levels, almost undetectably low levels, but nonetheless, you'll have all the intermediates in the pathway. And that solves a lot of problems, because it means that when a gene does arise, then all it actually needs to do is increase the slowest step in that pathway. And so it's easy to see then how the system can get better over time and how genes can improve that system. Whereas, again, if you have one step at a time, one enzyme arises and it catalyzes step 8, but you don't have steps 1 to 7 or steps 9 to 12, what use is catalyzing step 8? If all those other steps are there, then there is a use in catalyzing step 8, especially if that was the slowest step. So, there's ways of seeing it, and the big question here is, do all these pathways really spontaneously happen? And the answer is beginning to look like yes, they really do.
Can I just play radical extremist advocate here for a second? Abiogenesis.
Yeah.
Must it happen? Is it possible that we could just have panspermia eternally? Is the origin of life absolute? Yeah.
I mean, there's
just because science is so.
Panspermia is it's not actually helping. It may be true. The thing is, we'll never know how life started on Earth because it's a historical question. We don't have a time machine. And even if we did have a time machine, you go back 4 billion years, where are you going to go? Are you going to go to a deep sea hydrothermal vent or a Darwin's warm pond on land? Or are you going to go to coacervates in the atmosphere? Or are you going to go to a beach? I mean, if you see some green slime there, do you think that this is a stepping stone on the way to life? Or was it a failed experiment? Or is it already alive? Or is it a different kind of life that you never knew about? Actually, what we need is an intellectual framework that says, here are the steps that will go from an inorganic, sterile planet to life, either as we know it or as we don't know it. But what are the steps? And if we want to define life as we don't know it, then what actually is that? It's easy for me to imagine with computers or something, with programs, that's lifelike in many ways, but that's a humanly created system. If it's some other form of life somewhere else, how do we even begin to define it or think about it?
Or maybe
panspermia. So just to come back round, the problem with panspermia is we have no idea where it comes from, what were the conditions, how long ago it was, how long it took. There's no way of constraining it. So it's not science because it makes the question further away and it doesn't give you any experiment.
Yeah, that's such an interesting blind spot of science. Like, we obviously love science, but it only has the ability to make statements about that which it can know. And so even like you threw out this 4 billion year date, and it's like, well, that was definitely the last time the planet was resurfaced. I mean, that was the last time that fresh uranium was incorporated into the rocks that we have, which are very few and far in between to begin with. And so we don't know. Things we don't know. Like, we can't say, we see that life is here. We see that life doesn't really seem to come out of nothing every day. We don't just have like a new organism popping up in the bathroom in the morning all of a sudden that never existed before.
Maybe.
But still, we're convinced that it must have happened, which is interesting to me.
Hold on. I want to add something to that because
So inflammatory.
It is inflammatory, but I think that because this is a philosophical argument that we have all the time, right? Because like I remember the first time that Shiloh was like, "I think that life is eternal," and I was like, "You can't say that."
I'm not sure I said it quite like that.
I don't think so. I
think I just said it was possible.
Yeah, exactly. And so, and for me, I'm like, okay, maybe it is, right? Like maybe it is simply just when you have the right environment of thermodynamic gradients in a membrane, and it'll just kind of arise. Maybe it arose once somewhere far away, and it has been passed along eternally, or maybe it's constantly arising everywhere all at once, and it's just this thing that happens, and it's lost in the noise of there being other life.
Yeah, like, is there life? Do you think life's happening right now? It's just getting swallowed up by other life before we...
No, I don't think so.
Okay.
I think certainly my own take on the origin of life requiring hydrogen and CO2 and charges across membranes and catalytic iron sulfur minerals and things like that requires the strict absence of oxygen. And so 4 billion years ago, fine, there almost certainly was next to no oxygen. Probably for the first 2 billion years of life on Earth until the Great Oxidation Event. The geologists can't agree. There may have been, people talk about whiffs of oxygen 3 billion years ago. There's at least one geologist who thinks there was oxygen around 3.5 billion years ago. So I think consensuses are dangerous things in science, but insofar as there's a consensus, it's that there wasn't any oxygen 4 billion years ago, and there wasn't really any until about 2.2 billion years ago, and then we have the Great Oxidation Event. And I've discovered by misfortune as much as anything else, we've been trying to get hydrogen and CO2 to react in the lab in microfluidic chips and bigger reactors and all kinds of things. And for a long time, we couldn't make it work. And one of the issues was that we had an old anaerobic hood, which was perpetually a bit leaky, and we were patching it up, and we could measure the oxygen content inside. And it was usually between about 2 and 10 parts per million of oxygen in there. And I kind of thought, oh, well, that's pretty low. That's all right. Anyway, eventually this died completely, and we managed to persuade UCL to buy us a new, nice anaerobic glove box where we could get the oxygen levels down to less than 1 part per million. And suddenly all our experiments work. We're making organics much more readily. So what have I learned from this? It's that even trace amounts of oxygen kill this chemistry.
Oh, there are places, though, right? Like deep hot biosphere places where life is presumably flourishing.
But the deep hot biosphere has a really slow turnover, which is to say it's substrate limited. There's hardly any source of energy down there. So they persist. I don't know how it's measured, but
they're breathing rocks and doing all sorts of crazy...
about 1 million years for a bacterium to replicate itself.
What were you going to say?
I was just going to say they breathe rocks, and their substrates are not anything like the ones we find up here on the surface, which is quite interesting.
Well, it's still electron transfer. You're still dealing with an electron donor, which may very well be a rock of some sort, and an electron acceptor, which again can be a rock, but you're often dealing with trace amounts of hydrogen gas, passing it on to oxidized iron or something as an electron acceptor, that kind of thing.
Mm-hmm.
So the model for the emergence of life that you propose in the Vital Question is at a very specific kind of hydrothermal vent at the bottom of the ocean, where it wasn't the black smokers that are very aggressive, it was these white smokers that have a gentler flow of water through them, and that have this kind of perforated structure where you have inorganic membranes that hold these these iron sulfur clusters, basically.
Yes, and there is differences in proton concentration across the barriers as well. Not necessarily much in the way of charge, but differences in pH. And actually, the importance of pH is that I mean, technically it modulates reduction potential, and to put that more plainly, it effectively, if hydrogen is in alkaline fluids, it's more reactive. It's more likely to dump its electrons onto anything else. Whereas if CO2 is in acidic waters, it's more easily reduced. It's more likely to pick up electrons, pick up a proton to balance the charges, pick up another electron, another proton, and so on. So it's much easier to dump electrons onto CO2 in an acidic environment. And cells work pretty much that way. The proton gradients across membranes are in some of the proteins. They're effectively, it's modulating a redox switch to make for electron transfer from hydrogen to CO2. It's not it's not an easy process. It doesn't. The first steps do not happen spontaneously. Everything else does after those first couple of steps. So as soon as you've broken that barrier down, everything flows. But the first couple of steps are the hard ones, and what it's doing really is using protons as a switch to increase the reactivity of things and make them react. And it's that structure that you have in these vents. There may be other environments that have an equivalent structure. I'm not kind of necessarily tied that it has to be here and it cannot possibly be anywhere else. But what is beautiful about it is that you've got this cell structure. I said it was equivalent to the planet itself. It's like a battery. The outside is positive relative to the inside. And that charge which is coming in, or the protons that are coming in, are powering a redox switch that makes hydrogen react with CO2. So it may not be right, but it is rather beautiful. And in the lab, it does work. It doesn't mean that's how life started, but it does mean that the idea is not completely stupid or unworkable.
Mm-hmm. So do you secretly hope that maybe in the vents of Europa or something we're gonna find something like this happening?
Yeah. I wouldn't say even secretly.
All right. Fair enough.
I would say possibly Enceladus even more likely than Europa, but I think I'd be quite disappointed if we didn't find life there.
Why Enceladus?
But I would expect it would be bacteria. I wouldn't expect to find giant squid down there.
It just because it's so far from solar radiation that powers metabolism on Earth, or the primary producers of Earth, I mean?
Yes, it's partly that. I mean, the energy available would be limited, but it's also this thing we touched on earlier on about the history of life on Earth, that eukaryotes only arose once, and it would be very easy to imagine a planet where it never happened. There's no trajectory about evolution that says, okay, once you've got bacteria, just let the clock roll forward and you'll end up with humans every time, or you'll end up with this, sophisticated equivalents.
I thought you were advocating that at the beginning of the interview. Sorry, I misunderstood something.
No,
I see.
So
you think it would? I see. So you think the tape would play out differently?
It's more or less inevitable that we'll get bacteria. I mean,
but not. I see.
Embellishing here, but a much lower likelihood that we would see complex life.
Just because there's less factors at play in the beginning versus down the road.
Because I think a lot of the chemistry, starting with CO2 and hydrogen, is thermodynamically favored and will get you as far as the beginnings of genes. And as soon as you got to the beginnings of genes, we've just had a paper on this recently, which I mentioned in Transformer, but not in really any detail. But there are patterns in the genes, which is sometimes called the code within the codons, that suggest that there are biophysical interactions between amino acids and the anticodon that is associated with, that is cognate to that amino acid. These patterns are stronger if we kind of structure them around a metabolism starting with CO2. Then the patterns, instead of becoming weaker, become stronger and more striking. And we've been trying to measure directly in the lab. Do we see, using molecular dynamic simulations and NMR, do we actually see, does this amino acid spend more time with this nucleotide or more time with that one? And it's very probabilistic. They don't spend much time anywhere very much. But there's a tendency that the cognate amino acid will spend more time with the correct nucleotide than the other ones do. It's a kind of a statistical likelihood, but it's a weak trend. But it's enough. As soon as you've got that, then you've got you've got the possibility of a non-random code, which is to say, you can have a random string of RNA with a completely meaningless set of letters in it. If the amino acids that effectively bind to that are not random because there's a biophysical interaction between them, and they then zip up to form a peptide, there's a lot of asks here as well, then what you would have would be a random RNA sequence gives you a non-random peptide that can have function in the context of a protocell or a proto-metabolism or whatever it may be. And so effectively you've just pulled information out of a hat. You've now got something which can be selected for on the basis of that sequence. That sequence has meaning because it codes for a peptide which has a function. So although mathematically the sequence had no meaning, it does in the context of biology.
Well, the function of the peptide, I guess that's the big ask there, right? Because, I mean, if you have a nonsensical strand of RNA and it'll produce a peptide, the likelihood that the peptide actually does something, so I guess it makes sense to ask, like, where are you in terms of your own experiments and where do you think that they will be able to go? Because the idea that, yeah, let's start there. Go ahead.
I just wanted, you said something really fascinating before about, you said something about Enceladus being more favorable to this situation than Europa, and I was hoping you could elaborate on that just briefly before we talk about your lab experiments. Because I'm actually lecturing on the moons this week at the university, and I think all the kids are really, really interested in these topics of, well, obviously all of us are, of where would life be next? Could you just speak to that?
So the plumes on Enceladus, I mean, I think there are plumes as well on Europa, but the plumes on Enceladus, because Cassini flew by and took measurements of it, we know that they are alkaline and around pH 9. We know that there's plenty of hydrogen and there's some methane and there are other organic molecules in there. And that suggests that the oceans under the ice are also alkaline, which suggests that the most likely process to give you alkaline fluids with hydrogen gas and organic molecules would be serpentinization. So this process of rocks like olivine, and olivine again is really abundant. So it would be no surprise if there was olivine down underneath the oceans, in whatever you call it, underneath the oceans of Enceladus, reacting with the water. And producing alkaline fluids with hydrogen gas, the same kind of things that we know happened on Earth, and some suggestion of it on Mars, and probably happens on Europa, but we have less direct evidence on Europa.
Yeah, we're going to be back there this next year. Yeah. I think we're going to be back there this next year, 2023, 24. There's going to be 2 passes. I believe the orbiter is in this strange trajectory where it's like orbiting the poles of Jupiter. But it would be really wonderful if we could have the same hope about Europa, because it's much, much easier to get to. So.
And there's one thing which worries me about Enceladus, which is that if the oceans are alkaline, then we would have. I'm always thinking about gradients. I'm thinking about protons outside, alkaline inside, flux of protons in. If the oceans are alkaline, then we don't have much of a gradient. We don't have many protons around. We have a potential kind of proton deficit. Perhaps it doesn't matter. Perhaps it's become more alkaline over the last few million years or hundreds of millions of years. Perhaps it was more acidic a while ago. I don't know. But if the oceans of Europa are more acidic, and there is some evidence that there is serpentinization also happening on Europa, then we have a situation that's much more like the early Earth, as I imagine it, where we have an ocean, we have serpentinizing seafloor producing alkaline fluids, and then hydrothermal systems, which are alkaline with hydrogen gas, probably CO2. And so then we have an exactly analogous system. The reason I've said I favor Enceladus is that there is evidence at the moment for serpentinization and for hydrogen and for organics. But if that evidence is also there on Europa, that would be the favorable place.
Yeah, again, we can only speak in science to what we actually have something concrete to work on. It doesn't necessarily outlaw the possibility that other things are happening, but Well, it can do that, but not in this case.
Yeah. I mean, I think imagination is a really important part of science. But if it becomes only imagination, then it ceases to be science. But science without imagination, it really ceases to be good science, I think. So it's a strange balance. But I think, when you're touching on panspermia before, it may be true. Historically, it may be true. But we can't ask the question in a scientific way because there's just no way of approaching it. It's just, it's just imagination. It doesn't mean it's wrong, and it doesn't mean it's not wonderful. And, I love the idea that the universe is full of, Star Wars or Star Trek or something. I grew up with this stuff, and I really wish the universe was like that. But, everything that I've done as a scientist says, well, no, it's more difficult than that. And I imagine we'll find bacteria readily, but all these fantastic aliens that we want to meet, some people say they're out there and they're mixing with us all the time, and they're quite serious about this. And I tend to reject conspiracy theories as a kind of just as a reaction. Maybe I'm really stupid and they are there, but
maybe they were there a long, long time ago. And maybe I think it's just important to remember, as lovers of science, that science is inherently bounded by what we know for sure, and that it's really important to remember that as we learn more, those ideas will change and they'll expand and so forth. So all of the possibilities are just overwhelming. It would be a complete imaginative chaos if we just allowed our minds to run free. But it doesn't.
I think we need to remember how powerful science has been in getting us to where we are and interpreting the world and so on. So you can say it's a failure of imagination on my part to think about life in terms of life on Earth and being carbon-based and being water, and that may be true. But what I'm trying to do is apply the principles of science to it, which is to say, let's try and understand how this system works, and then we can ask, well, could it have worked a different way? Is there a really good reason why it works this way, or is it actually just an accidental reason and there's 10 other ways that it could have worked? With an N of 1, it's the only way we can actually scientifically ask this question. Otherwise, it's pure imagination.
So then let's talk about what it is that the specifics of what you're doing in your lab. What are you have this anaerobic glove box where you're setting up experiments in the hopes of what would be the holy grail of what you're aiming to show?
Well, there isn't a single holy grail. I mean, there's this long continuum going from the simplest prebiotic chemistry, and here the experiments we're doing is using microfluidics to have effectively two phases in laminar flow parallel to each other that precipitate a barrier between them. So a very hydrothermal kind of system. One side alkaline with hydrogen, the other side acidic with CO2, and we know the pH gradient makes the difference, and we know that it can form organic molecules. Simple ones, formate, acetate, maybe pyruvate, things like that. So we're getting, to the Krebs cycle. We've not published much of this yet, but we're working on that, and it's fairly robust. So that's one aspect. Those are the first steps, going from CO2 and hydrogen to the simplest organic molecules. Then, as a separate set of experiments, the question is, well, if you've got Krebs cycle intermediates, can you make the amino acids? Can you make the nucleotides? Following the biological pathways, can you make sugars? Can you make fatty acids? So it's all being done by the chemists, but with different starting points usually. So the question is, the biological pathway that has 10 steps in it, which is conserved in bacteria and archaea to make purine nucleotides, for example, can do those steps happen spontaneously in the absence of enzymes? And, the answer is no. But the question is, okay, what about a metal ion catalyst? What about, let's try these 10 different metal ion catalysts. What about pH? pH probably matters. Does it work at pH 4? Does it work at pH 6, 8, 10, 12? So what about temperature? Are we looking at 0 degrees or 20 degrees or 30 or 50 or 90 or 120? What about pressure? Does that matter? So, a very simple experiment, it balloons out into a whole series of possible conditions. And one of the things that we try to make sure we do is not believe our own hypothesis. We try to be agnostic, I guess, about the conditions. So we just try to find any set of conditions under which these things might work. We look for the steps that are the most. Very often you see similar chemistry at multiple steps. And so the question is, if you optimize the chemistry at one step, does that then work for the other steps as well? And if it does, then you're beginning to think, okay, so what about the steps that don't work that way? Can they work under similar conditions? So you eventually try and find a set of conditions that will work for the whole pathway. And often it turns out that we're dealing with a fairly limited range. It often works between pH 5 and 9, say. It's frequently working well at 50, maybe 70 degrees centigrade. High pressure doesn't seem to make such a big difference to most of this chemistry. The salinity matters sometimes, not other times. It's hard to know. So there's all, the metal ions, they really matter. They matter more than anything else. And we keep finding it's the same ones. Iron is really important.
Are you surprised that pressure doesn't have an effect?
It has a small effect. So years ago, when I first started doing this, and I'm talking 15 years ago now, I was talking to some guys in the chemistry department at UCL, and they said pressure. It's got to be pressure, Work on, and I eventually I backed away from it because we were trying to build our own reactors and building a high pressure through flow reactor. I wanted it to be like a vent where we had a continuous flow in and flow out. And to do that at, say, 100 bars pressure is quite an engineering feat. Yeah, and I'm not an engineer, and some of those guys were, but it was much easier to build a pressure bomb reactor where you have a very high pressure, but nothing comes in and nothing goes out. And that's not what I was looking to build. And the real reason why pressure might make a big difference is the solubility of hydrogen gas. And for a period, I thought, if what hydrogen is doing is dumping its electrons onto a barrier, and that barrier is composed of mostly ferrous iron, say, and then the electrons pass on to CO2, so the iron becomes oxidized again, then the hydrogen can pass electrons onto the ferric iron, reducing its ferrous iron, pass the electrons onto CO2. So effectively, the original idea was electrons cross this barrier, and the barrier is therefore catalytic. Well, perhaps you don't need the hydrogen. Perhaps if you just have a barrier which is only made from ferrous iron for a period, there's plenty of electrons on the iron and they can pass on to the CO2 and we should see exactly the same thing. Then we don't need the high pressure because we don't need to dissolve hydrogen, and so it makes it much easier. None of it worked. And it turns out you really do need hydrogen in this system. If you don't have hydrogen, it doesn't work. And the real reason, and now we're trying to pressurize our chips because the higher the pressure, the more hydrogen you have, and the greater your driving force, and the better everything will work. But a lot of the rest of the chemistry, the pressure doesn't matter so much.
So the reason that I'm interested in the pressure question is that we've recently been looking into the idea that the Earth used to have a much thicker atmosphere. So there's all these mini Neptunes that they're starting to discover, where they basically think that they are this intermediate between the gas giants and the rocky planets. And so they have these very heavy water vapor atmospheres that, as they move into the center of solar systems, are photoevaporated, leaving behind a super Earth with this like 4x the size of Earth rocky core. And we were talking a while ago, there's this NASA astronaut, his name's Don Petit, who, you want to tell
some of the super Earths don't have to be that big. They can be Earth-sized. Yeah.
Okay.
They're called super Earths, but they can be Earth-like.
All right, ambiguity. No, that's good. That's good. Do you want to tell the Don Petit story?
Not at all. Yeah.
So Don Petit, when he was a grad student, was a mechanical engineer, and he was working in a laboratory with a PI who was kind of one of those guys who really liked riddles. And so they started to look into the question of how it is that pterodons could fly. Pterodons are these massive reptilian dinosaur. They're not technically dinosaurs, but we're not going to get into the depths of the dinosaur classification. He was like, look, they're too big. Like there's this mechanical engineer named Tanaki who put together what's called the Great Flight Diagram, which is a relationship between wing loading and wingspan. And so the size of the animal is limited by the physics of the atmosphere. Like a creature that is too big cannot fly. And these pterodons are too big to fly. And so the question is this: were they truly just gliders, and they had these enormous wings? And I mean, the way that people have them is that they're these massive birds that would have to clamber to the tops of cliffs and then like glide off of them, and it's absurd. And so their proposal was that the atmosphere was thicker,
Not even that much thicker,
just
right?
like 3x.
Yeah.
I mean, we're certainly losing atmosphere.
Yeah.
I mean, this goes back quite a long way. The idea. So I wrote a book 20-something years ago called Oxygen, and it was about. Sorry, you there? Yes, it was about the oxygen content of the atmosphere, and there was a guy called Robert Berner at Yale who had. More or less calculated based on rates of carbon burial and all kinds of fairly abstruse factors, but they were very solid, that the atmosphere had about 35% oxygen in the Carboniferous period, which corresponded to a time when there were giant dragonflies around with a wingspan of nearly 1 meter, so 3 feet. And I never worked on any of that stuff, but it was fascinating. And probably 35% was an overestimate. It's gradually crawled down as these analyses have got better, and I think now it's around 28%, but it's still substantially greater than today's 21%. And so this is much earlier than pterodons, and people have tried. Then what happens if we breed dragonflies over, say, 10 generations at high oxygen levels? Do they get bigger? And the answer was yes, they do get bigger. Not that much bigger, not to 3 feet wingspan, but they increase their size by 20% or 30% or something. So there's, even in a short number of generations, they changed. And the reason was an interesting one. It turned out to be not to do directly with the flight mechanics so much as the tubes, the trachea that are providing oxygen to the flight muscles. So they don't have capillaries or anything. They've just got these open tubes. And if you have a higher oxygen concentration, the tubes can be narrower and you get the same oxygen to the end user tissues. And because the tubes are narrower, then you can have more connective tissue between the tubes, and that gives you stronger anchoring for the wings themselves, which increases their mechanical properties, and that allows them to lift a heavier insect. So it was not actually the reasoning that people originally thought, but it's partly true that higher oxygen levels. But if we go back much further, 4 billion years ago, we have a problem with where did all the CO2 go? And the atmosphere back then was probably at least 10 bars. It's hard to know. It's hard to constrain. I don't think that there's any suggestion that there was 3 times the atmospheric pressure around the time of.
Well, what's crazy about that question is that there's no, the partial pressures would be the same relatively. And so it's not as easy as looking at the chemistry in the past, right? The amount of oxygen relative to other gases would still be the same. It's just that the pressure of them would be higher. And so that's an extremely difficult thing to pull out of the record, other than by making inferences from things like dinosaur flight. Of course, it's totally possible these were just like giant flying squirrels or something, but it's a compelling idea still. And it seems to be in line with the more that we learn about how planets form as well. That certainly our planet is losing atmosphere at an appreciable rate, and if you extend that trend backwards, it's hard to ignore the fact that it must have had more atmosphere at one point in time.
Oh, for sure it did. Yes.
But the partial pressures could have been the same, effectively, relatively speaking. So
when you test in the laboratory, you say that you're putting the chips under pressure. How much pressure are we talking?
Oh, very little. Less than 5 bars.
Interesting.
Actually, a lot of the time we're just working still at atmospheric pressure. There's a former student of mine, Eloy Camprubi, who built a high pressure rig for microfluidics that operates at 70 or 80 bars. And he's just moved to Texas recently, so I don't know if he's got the thing set up and working again now. But I think he will do some fantastic things with that.
Interesting. We'll have to. What was his name?
Eloy Camprubi.
Okay, we'll check him out. Let's see. So you've developed these reactions. You have shown that there's spontaneous production of certain intermediates of the cycle. To you, it speaks to the metabolics first? Would you agree with that?
Yes, I mean all of that does. By no means have we proved any of this, because what we're really looking for would be the entire metabolic network happening spontaneously. What we actually have is a few steps here and a few steps there, and it's piecemeal and it's splintered. So it's a long way from being able to say with any confidence that this is true. But at the same time, there's enough bits here and there to make you think, well, it's not completely wrong either. So, let's keep exploring. I mean, that's part of it. But then there's other aspects like, what about the protocells? If we can make a bilayer membrane with fatty acids and very simple amphiphiles, are they robust under these conditions, under alkaline hydrothermal conditions? Yes, they are. Can we make the iron sulfur clusters? Can we collate them even with single amino acids? And yes, we can. And we can just use cysteine alone at alkaline pH, and you get the 4Fe 4S clusters directly. So there's lots of, I mentioned where there's another PhD student who's looking at these interactions between amino acids and nucleotides based on the patterns in the code, which again is a mixture of lab work with NMR and molecular dynamic simulations. And we're trying to do other modeling work to try and just work out what's the likelihood of these things happening, which direction should we look in, what kind of things ought we to be thinking about. So there's a lot going on. But there's such a long distance to get to. By the end of it, we need to be on to ribosomes, molecular machines, conformational change. This is the end point. By the time you got to a ribosome, I think you can say we've got something which is pretty much alive now.
Mm-hmm.
It seems possible. Oh God! But you'd have to get there with no external inputs, right? Like that's
Yes.
you're trying to get there. So from CO2 and hydrogen to a ribosome
In a structured environment.
in a structured
With a
environment.
continuous flow of fluids and energy and hydrogen and CO2 and iron, and this is a very dynamic environment, and it's far from equilibrium, and you get the structures for nothing, and they organize themselves into protocells. Again, you get the spontaneous membrane structures forming. They tend to bind to mineral surfaces. There's a lot of structure you get for free in these environments. This is not a soup. I get very irritable when people I say, "Oh, so what about the primordial soup then?" And this is structure and flow, and charge and organization. It's all just part of this system. It's all part of the giant battery.
What are the biggest molecules that you've gotten out of the system?
We're not really kind of starting at the start and trying to get to the end. We're taking bits here and bits there. So it's, we do some experiments with larger molecules and some with smaller molecules, but I don't think we'll ever kind of just feed in at one end and watch come out of the other end cells. I don't think that's ever going to happen. Not in my lab. It's not the aim either. Some people may say it's their aim, and I think we're a long, long way away from that.
I mean, I think that it's in order to be a really, really convincing story of the origin of life, you kind of you have to be able to do that, right? Do you agree or no?
Yes, I mean that's a Feynman kind of quote, isn't it? That if you don't understand it, if you can't make it. We're talking about making the most miraculous machine in the universe. Basically, it would tax even Feynman to do that. There's an awful lot of steps. I mean, I think the other thing is a matter of scale as well. So we're talking about the entire seafloor probably, because the entire seafloor, there's a fair amount of evidence was pretty mantle-like, which is to say is covered in komatiite lavas and things like that, which are full of minerals like olivine. And the crust and the mantle hadn't really differentiated, which means that the stuff that was directly in contact with the ocean was pretty much like the mantle, and therefore was going to react with the water and form these vents across the entire seafloor of the entire planet. With a range of chemistries going on, depending on what's actually the rocks down below, which minerals are coming up, and we've got as many millions of years as we want. I don't think we want millions of years, but maybe we do. But in any case, we have them available to us. So the planet is a laboratory over millions of years. So the idea that we can, with a few PhD students over a few years, go in a lab and make all this happen, it's not going to happen. If we did a kind of a worldwide Manhattan project on it and really made, a serious effort to do it on a grand scale, perhaps then it could happen. But at the moment, nobody would agree about the conditions that we should use. I'm in a minority. There's a few people like Bill Martin and Mike Russell and so on who think in this way about, my own inspiration is coming directly from them, about these hydrothermal vent systems and so on. A lot of the chemistry that's being done now, there's a guy called Joseph Moran who's done a lot of really beautiful chemistry showing that this metabolism really does happen spontaneously. So there's really a handful of people in the world who are thinking about the problem this way. Most people are seeing it in different environments, different circumstances. If anyone gets the funding to build a Manhattan project to simulate the origin of life, they're going to start somewhere else with different starting points, and in my view, it's going to fail.
I mean, is the goal to... Is it a philosophical goal for you more than it is an engineering goal?
I would just like to know, how does life start? How does a sterile, inorganic planet give rise to this amazing living planet that we see around us? Is it easy? Will it happen anywhere? Is it really difficult? And what a wonderful thing that the human mind, regardless of whether it's an engineering problem or a philosophical problem, can understand, in principle at least, how it could happen. And we can't pretend to know how it happens until we've done an experiment effectively at each and every step, which shows that, yes, this step is realistic, and the next step is realistic, the next step is real. To put all the steps together is a scaling up problem of a completely different order to anything that humanity's ever done before. But to demonstrate that the steps themselves work is a feasible undertaking.
It is certainly a project aimed at understanding who and what we are too. At some level, it's like, what is this? What is this? What does it mean? Like, we didn't ask you earlier. We probably should have, since we've been talking about life this whole time. But what is life? Do you do you have? This is certainly something you've thought about a lot, and actually we think about it all the time. We just had Lee Cronin on the show, and we spent a great deal of time trying to circle around this idea. Is there a succinct statement that is unambiguous and consistently applied that you can put behind the definition of life? No. Forget it.
Because Lee Cronin's idea is that life is that which produces complex molecules in abundance.
I mean, that's probably true, but it's not a definition.
It's a thresholded definition.
That's a black box. It says what it does, not how it does it.
Well, all of our definitions of life are kind of like that.
But I mean, I agree with that. There's an ambiguity that geology and geochemistry can give rise to quite complex molecules, but life gives rise to much more complex molecules. And if you want to, he thinks about non-carbon based life, and that's why he has this system. Because if we find really complex molecules that are not based in carbon somewhere else in the universe, can we have some objective measure which says that, well, this is too complex to have formed by any kind of planetary chemistry that we have any knowledge of? And he's teamed up with Sarah Walker, I think, on this kind of, and it's a very simple and smart and clever idea, and I like it. I frankly would be very surprised if they find very much that's not carbon-based of that level of complexity out there in the rest of the universe. But I'm very happy to be completely wrong. I would, it's one of those things where I'd rather be wrong than right. But I'm skeptical. But in terms of definitions and so on, I find it a really unhelpful way of seeing the question, because there's a favorite quote of mine from Peter Mitchell, who was the guy who first put forward the chemiosmotic hypothesis, the idea that there's a difference in proton concentration and charge on the two sides of the membrane. And he was at a conference in 1957 in Moscow about the origin of life. And there's a quote from his talk at that conference where he's saying you can't think about life without thinking about the role of the environment. I'm getting the quote wrong. I'm just giving you the gist of it. And that if you think about life, and he's thinking about cells like bacteria, then it's two equivalent phases, which is to say the inside of the cell and the outside world, separated and linked by, with a membrane that separates and links them, the two phases. And that's not a definition of life. That's a definition, that's a kind of a system, which is life is part of it and the environment is part of it, and it's treating them as equivalent phases. But if you think about the origin of life, and I've got a barrier and I've got phases on either side of the barrier, and it's the dynamic equilibrium across the barrier of things crossing that barrier that, and the driving forces involved that are giving rise to life. So the environment is as important as the thing that becomes the cell. And actually, without, I think NASA have a working definition of life, which is something along the lines of a self-sustained chemical system capable of undergoing evolution or something like that. And I really react against the self-sustained. Life is sustained by the environment. And of course, they're smart people. They know that. They're emphasizing that it's capable of generating copies of itself by exploiting the environment. I know what they mean, but they're not exactly emphasizing the importance of the environment. So I think in phases, and I think in terms of processes over time and change over time, and none of that is if you say what is life, that's a thing. And I would say it's much more of a process and dynamism over time, and so it's not easily defined as a noun.
I love how that actually gives us, I think the reason we ask these questions is so we can understand ourselves better. And I think that it's really important, especially in these exponential technological times that we're living through, that we do realize that we couldn't exist without the other pieces of the planet, right? Like I could not live without the trees outside of my window. It's not like a metaphor. It's literally true. We need them because they give us the oxygen that we breathe. So I love that you emphasize that. And I think that is a really important point, but it's also terrifying because you end up with you end up back into chaos all of a sudden when you realize that the whole universe is conspiring for the production of ourselves. It's like very difficult we want to be able to just peel off these little pieces and draw the lines between different objects. And it's not always such a simple thing to do. It's kind of remarkable that we
classification system we are prone to, and that's probably an evolutionary thing. Is that a tiger or isn't it a tiger? It's a bold question that we need an immediate answer to, and we try and do the same thing with almost everything. Is this a mineral or is it a living thing? Is it gonna poison me or is it safe? We need to categorize. It's part of deep human nature. But it's actually unhelpful when we're thinking about processes that change over time. And things that evolve and as I say, we're changing from a system which is basically just doing simple chemistry to something which is making molecular machines. And there's no point at which it becomes alive where you can draw a line across this continuum and say, now it's alive. Zing, it's that moment there. It's not like that. It needs all of this context and all of this continuity. And it's a long process over, I don't know, I'm tempted to say billions of picoseconds or something. Chemistry is fast, amazingly fast. We have a large supply of it, as much time as we need. But I think with chemistry, it has to happen quickly.
But the evolution of the conditions under which that chemistry can occur might take eons as well.
Well, to get to molecular machines on the scale of a ribosome that makes proteins, 200 amino acids per minute or something to build proteins, this is a level of sophistication. But as soon as you start thinking, well, what were the first molecular machines and how did they operate? And you think about, well, okay, I've got a peptide and it's got two states that it could fold into. They're metastable states. And is there a set of conditions that will flip from one state to the other state? Something binds here or whatever it may be. And, the concept of a molecular machine is not very difficult. The reality of a ribosome is mind-boggling.
Yeah, the ribosome is the final frontier. That seems like that's the moment where something very special is happening all of a sudden.
Well, I feel like it's the first real actor. All right, it's the first, the reactions, if they're happening spontaneously, and the association of the codons and the anticodons, if they're also kind of spontaneous, there's this glob that comes in and that can force things into position and and is sort of the first. It's the first multi-tool.
Communicating almost.
Kind of, yeah. Like it does seem like it
with
has
the other structures. I don't know.
Yeah, it's a node in the network, right? It's a node that basically brings all of these things together. Catalyst.
I think by the time we have things like ribosomes, we also have genes. Genes are kind of easier. Not necessarily a complete genetic code and not necessarily a fully functional system, but strings of RNA capable of producing cognate peptides that's plated onto it in some way, giving rise to peptides that can have different ways of folding and different functions in the context of a growing protocell. If it's a hydrophobic peptide, it'll go to the membrane. If it's sitting in the membrane, maybe it makes it easier for protons to come through and funnels them onto an iron sulfur cluster, which probably sticks to a hydrophobic peptide. There's there's lots of ways you can imagine things happening, and we can test those things, and some of them may be true and some of them may not be true. But conceptually, it's not so hard. Conceptually, how a ribosome manages to, what's the minimum possible set of moving parts that makes a ribosome? You need to identify the reading frame. You need to process along the RNA frame by frame without slipping too much. So you can tolerate some slippage, but you need to go this way and not that way. So what are the basic mechanics of this system? Cut out all of the ribosome and think, can I do all of this with tRNA? One tRNA or RNA, what are the minimum moving parts that I can make this system with? Now we're into more kind of engineering territory in a way.
And it seems like
RNA
that's
world territory.
Right, but it seems like that's kind of where the application of the origin of life research comes down to, because I can imagine this being, if humans are a spacefaring civilization, then basically what they do is they go to these planets that they want to terraform, and what they set in motion is they set up the conditions necessary for life. And if you know what those conditions are on a dead planet, and you can figure out how to tweak the environment in such a way where you can plant the seed and walk away.
I think the conditions for the origin of life and the conditions for life are not necessarily the same thing.
Sure. But like you said, it's
for life, we would want to have oxygen now. If we want to have plants and we want to have animals, then we want to have oxygen. But if we wanted to have oxygen, then we're not going to have an origin of life in an oxygenated world.
But even technologically, in terms of incorporating it into robotics and things like that, a journey to the next star system over is going to take, half a lifetime. And the spaceship's going to have to be, maybe the spaceships could be repairing themselves. Maybe we can apply these biological principles about origins of life to engineering interfaces that are capable of doing a lot of the magic that we can't even conceive of doing.
Yeah, it seems like what you're developing is the sort of the rules for self-creation. Of self-authorship.
Yes, I would say probably I would limit it to organic self-creation, which is to say, one of the nice things about CO2 is it's like a Lego brick. It's there in the atmosphere. You pluck it out, you stick it onto something. Whereas silicates, sand, they're huge, gigantic macromolecules at the level of the atoms themselves, and you can't build with sand in that sense. But I'm not sure you'd want to build a spaceship out of carbon. Or at least not out of organic carbon. So I think the principles might carry over, but the actual mechanics of it probably wouldn't terribly well.
One of our audience members, he's been writing biological sci-fi, where he's talking about he's taking the tropes of sci-fi and putting them through the lens of technology that is grown rather than created, and it's something that I've been thinking a lot about because we spend a tremendous amount of effort recreating the mechanisms that life has already figured out with much greater efficiency. And I always think about what a world would look like where instead of having to build things in this really artificial way that we do, if we could,
which is really inefficient and dirty.
Yeah, exactly. Like we have a very polluting way of producing goods, and if we can harness the way that life organizes itself and the way that life builds things.
Francis Arnold, who won the Nobel Prize for Chemistry a couple of years ago, has been working on enzymes, effectively selecting for enzymes that do chemistry that life never does. So really difficult problems in chemistry, using the processes of natural selection to select for specific enzymes that can do that chemistry, using very often the kind of metal cofactors and so on that are used in, say, liver enzymes or something that does lots of detoxification. But you can change them amazingly to do the kind of chemistry that really challenged physical or synthetic chemists to the point of not being able to do it at all or extremely inefficiently. You can do it really efficiently by bringing to bear the process of natural selection in a controlled way with a target in mind. I mean, it's a fantastic work.
And I think that it is if I think about the technological future, I can't help but think that the machinery of life that we're really starting to understand now is the basis for the next technological age. Like you look at the work of somebody like Michael Levin, and it's terrifying on one hand because it's opening the door to the manipulation of body plans and of even cells into these creatures that appear like they have a mind of their own. Like he seems like he's creating life from in like almost panspermically, right? You have these frog skin cells that you can like.
Yes, I mean I wouldn't see it as a very threatening form of life, but who knows where it can go. But I think what Mike Levin is doing, which is extremely valuable, is breaking down the kind of deep conservatism among biologists who will see development as purely a genetic program. And if you start talking about electrical fields and so on, we'll back away. And you have to have exceptional quality work to get people's attention and make them take it seriously. And he's doing that work. And you know, it's really beautiful. It doesn't surprise me very much. It's very surprising in some sense, but in another sense, I've been thinking about mitochondrial electrical fields and electromagnetic fields and so on, and what work can they do? And he's been thinking about the plasma membrane of cells and the bioelectric code and so on, which is not something I know much about or have thought much about, but the idea that you can organize things with electrical fields is very appealing. Clearly, it's true. It's almost certainly true for us as well in almost any organism. I think he's bang on looking for a bioelectric code. And it's just a whole open frontier of biology that almost nobody has gone anywhere near it. And, you know, it's going to be, I was going to say, as important as genes. Maybe not, because without the genes, you don't have anything else. You don't have the structure or anything. But still, it's a hugely important area of biology that is you know. I ask lots of evolutionary biologists or evo-devo people who work on development and so on. Are you familiar with this work on bioelectric fields? Hardly any of them are. It just doesn't kind of cut through to mainstream biology because it's so conservative.
And that's kind of the feeling I got by going through biology to the highest levels of it, right? You go through the PhD and you realize that it is fundamentally a very conservative field that still feels informed by the way that experiments were being done in the 1950s. And we haven't really gotten into the bioelectricity stuff, but there's a lot of really odd research that has percolated through the field over the course of the last 30, 40 years that has been outright dismissed as being charlatanry or nonsense or whatever else you want to call it, that it really seems like what Mike Levin is doing in the idea of bioelectrical codes in life is
it would also be dismissed as mumbo jumbo, except that his work is so good.
And that's the crazy thing. You make something that works and people cannot help but be like, well,
Well, I mean, you believe the evidence of your eyes. That's exactly what he shows you. He gives you the evidence for your eyes, and that's why he is taken as seriously as he is, and as why he's made me sit up and think seriously about, well, how are the mitochondrial fields going to integrate with plasma membrane and development and all these other things and consciousness. So they're really big, interesting questions. But I think he's cut through to scientists and he's cut through to the general public, but he's not cut through, to my mind, yet to the molecular biologists.
Interesting.
Why do you think that how is it that he cut through to you? Because you're very you're molecular in spirit, but is it because you're focused on this larger question?
I've always been interested in bioenergetics and the membrane potential and the questions of what kind of fields will a membrane potential I mean, obviously a strong electrostatic field, but what we've discovered over the last 10 years, not me, but people in the field, is that the structure of Christie is, rather than being kind of wavy line that they're often drawn as, they're actually very tightly focused. And at the end, you have dimers of the ATP synthase, and you have a kind of a current of protons now. So an oscillating charge in effect, which will generate electromagnetic fields, which is difficult to measure, and we have parallel Christie with parallel fields, which should interfere with each other and ought to strengthen each other so long as they're oscillating in phase and so on. So this is, again, a whole area of biology that I'm increasingly interested in. Is not linked directly with Mike Levin's stuff, but you can see where the connections lie.
Yeah, you can see the horizon there. You can see how the next century could just be a whole revolution in the way that we think about biological signaling and communication and integrity, and it's so fascinating.
I mean, it makes me a little bit nervous because when we start talking about the importance of bioelectricity, I look around at all of the devices that we're surrounded by constantly and all of the electromagnetic radiation that we're putting out into the world. And, you think about sentinel species like newts or toads or something that live in polluted waters, and they're the first ones to get sick. And I wonder if there isn't a similar thing that might be happening to very, very sensitive species due to overly abundant electromagnetic radiation that we've kind of decided is relatively safe. But again, if we're not looking for it in these ways, and this is a new discipline and it's kind of the dark matter of our understanding, what do we have to know in order to be able to even figure it out?
Well, I mean, there's a body of work going back over, again, decades that externally imposed electromagnetic fields and so on can influence physiology, but not random wavelengths and so on. A lot depends on exactly how strong is the field, exactly where is it, exactly what's and so on. Which is not very surprising, because if we're dealing with the electrical fields that cells may be dealing with, we're in quite a constrained range, and with a very detailed structure of the cell, and it will fall away very quickly from the membranes themselves. So you'll have. I mean, I often say that the strength of the field with the mitochondria, if you were to the strength of the field strength of the electrical field is about 30 million volts per meter. 200 millivolts is across a 5 nanometer membrane, so 5 millionths of a millimeter membrane is 30 million volts per meter, which is like a bolt of lightning. So it's an extremely intense field, but over a very short distance. And to generate fields that operate over longer distances, we seem to be needing to move to electromagnetic fields rather than electrostatic fields. And that means structures, which should be predictable structures that we should be able to see and we should be able to measure them, but nobody's really trying.
That's interesting.
Well, get to work, youngsters who might, if anybody's in college and listening to this, that's a ripe field for investigation. I always try to structure my lectures with the interesting pieces that need to be done, because when I went through school, it was kind of like, wow, we basically figured everything out. There's like some details to fill in, especially in physics. It was like, wow, all the big stuff's been figured out. And I think it's a terrible disservice to all the young people. But
I completely agree. Yeah. No, I think in biology, almost all the work's still to be done.
there you go.
All the most exciting stuff, anyway.
Yeah.
What are you looking forward to most?
It's not easy to give a straight answer to that. I think what I enjoy most is the process of discovery, of finding things out, of talking with people about how to get at a question, and then doing the experiments and finding answers, and then finding that I'm confounded by the whole process of science of doing it. If the questions are exciting questions, and you're doing it with people who are excited about it as well, and that's the pleasure for me. So there's lots of things I would like to know more about, but I'm not going to delude myself. I'll probably go to my grave, and maybe it's tomorrow for all I know, without knowing the answer to half these questions. One of the lovely things about science is that it's the unknown. You're exploring the unknown, and if you succeeded into turning all the unknown into the known, then you probably feel a bit disappointed afterwards. So I think the journey is a large part of it, and that for me is the excitement.
It's beautiful.
Yeah. Wow. And you've contributed so much already. You've got 2 books out right now?
Many more
Many
books.
more, yeah.
5, yes.
Your book Vital Question was basically required reading when we were going through grad school. So this is and it's very technical, but I certainly think everybody should check it out. Yeah, wow, so much to think about here. Yeah, thank you so much for coming and giving your time to explain these ideas and discuss them and entertain some of our radical extremist ideas. And it's been a really amazing couple hours.
Yeah, thank you so much, Dr.
Been great fun. Thank you.
All right,
Pleasure.
Take care. Have a great rest of your day.
Bye now.
Bye.